Good morning, everybody. Let's begin with the chapter 6, Molecular Basis of Inheritance, module 2. In this, we are going to discuss about the experiments, Griffith experiment and Hershey and Chase experiment, then semi-conservative experimental proof for the semi-conservative mode of replication. So, we will go to the Griffith experiment first. So in the year 1928, Frederick Griffith, he performed an experiment to prove that in organisms, especially in bacteria also, there is a special capacity that by the influence of a particular component, it can transform from one strain into another strain. So that process he named as transforming principle. The bacteria can transform from one strain into another. So he conducted, in order to prove that process, he conducted his experiment in a bacteria which is called as Streptococcus pneumonia. And in that, if this particular strain is affected with, it is allowed to infect a mouse, then they will show the disease. But he noted that there are two strains in that particular bacteria. And based on the external appearance, he named it as the S strain, Streptococcus pneumoniae, S strain, and the Streptococcus bacteriae, R strain. So what this S signifies is he observed a smooth outer coating for this bacteria. This is due to the presence of a particular type of polysaccharide. So if that polysaccharide coating is very smooth, then he named it as smooth strain. And if it is rough means he named it as rough strain. So S strain and R strain. Along with that, he found out that S strain is virulent. So what do you mean by virulent? It can cause, if that strain is injected into the mouse, means it will show the symptoms of pneumonia and later it will die. So it is virulent. Whereas R strain, he noted that they are avirulent or non-virulent. They cannot cause the disease and the mouse will not show the symptoms of disease. So one variety, that is S strain, is virulent and R strain is non-virulent. And he injected the S strain into a mouse and he found out that the mouse were showing the symptoms and later it dies. Whereas when he inject R strain into the bacteria, then it was not showing any symptoms of pneumonia and it was survived. So this is the step. These are the steps how he has conducted the transformation from one strain into another. And in the second step, what he did, he heat killed the strains. So he took the S strain bacteria and he applied the heat. And that strain he named it as heat killed S strain. Then he injected into the mouse and the mouse survived. So from that he concluded that when the application of heat or by the application of heat, the material which caused the disease is getting destroyed. And that is why the mouse were able to were able to survive even after injecting the heat killed S strain. And in the second, along with that, simultaneously he did the heat killed R strain also into the mice, and there also the mouse survived. And the third step when he took the S strain, which is heat killed, which cannot cause the disease. But when he mixed that one with live R strain, and he injected into the mouse, and he found out that the mouse died. That means, even though the heat killed a strain, they cannot cause the disease. Their disease pathogenicity they cannot create, but that a strain, some material is capable enough to transform that R into S type. Not only that one, after from the dead mouse, he was able to extract the live R as well as the live S varieties. So from that he concluded. So when he applied the heat killed along with the live R, he concluded that from the heat killed S, some material is entering into the R strain, and that material is capable of transforming the R strain into S strain. And that S strain, live S strain, is causing the disease in the mouse, and that is as a result the mouse died. So this is the sequence in this figure. This is the sequence of his experiments. So you see, the first one, he has injected the live R cells, the mouse were able to live. And live S cell, when he was injected, the mouse affected with pneumonia and then it died. The third step, he took heat killed S cells and injected into the mouse. Mouse survived. The last step, he took living R cells. Along with that, heat killed S cells. Later, he found out that mouse got the symptoms of pneumonia and it got died. And when he extracted the cells, he found out S cells along with R cells from the body of the dead mouse. So S cell was not injected, heat killed S cells only injected, and R cells were live. So this, from the heat killed cell, some transforming principle or transforming agent is entered into the R, and that transforming agent was capable of transforming R into S type. Because of that, the S can cause the disease, and the mouse got the disease and it got died. But he was not in a position to explain actually what is the chemical nature of that transforming component. And he concluded from this, the presence of heat-killed S strain bacteria caused transformation of some R strain bacteria into virulent by a chemical substance that he called it as transforming principle. But he couldn't experimentally prove that one. Later, three scientists in 1944, that is Oswald Avery, Colin MacLeod, and McCarthy. These three scientists were able to give experimental proof for the transforming principle. And they concluded that the transformation is caused by the DNA that is present in the cell. So here the experiment, how they have proved how they have given the experimental proof for the transforming principle. What they did is they separated the components, because the cell itself is made up of three components: DNA, RNA, and protein. And they separated each component separately, and that they injected. The first step, they took the protein, then injected into the mixture of heat killed S cells as well as the R cells, live R cells. When they took the protein and injected, they could not show the transformation. The transformation was not taken place. The same way, the extract of RNA injected, they couldn't observe the transformation. But when they took the DNA which is extracted, injected along with the heat killed S and live R cells. They found out the they were able to find out the S virulent forms in the mouse, and the mouse got infected with the pneumonia and it dies. Then in order to prove further, so that from that they said it is the DNA which is causing the transformation. Then in order to further experimentally prove that, they have taken the enzymes. They took 3 enzymes, that is protease, DNase and RNase. The speciality is that protease can destroy the protein, RNase can destroy the RNA, and DNase can destroy the DNA. So when they took the protein digesting proteases and RNA digesting enzyme RNase, and they found out that they couldn't transform that treated cells, that is DNA, protease and RNase treated it killed S cells with live R cells. They were not causing any transformation. They were able to transform. But when they took the extract, that is the DNase enzyme, into the mixture of it killed the cells and S cells and R cells, live R cells, they found out that DNase enzyme can destroy the DNA in the S cells, heat killed S cells. As a result, the DNA is completely destroyed, and that destroyed S cells cannot convert the R strain into S strain. So that means they inhibited the transformation. So the DNase can, since the DNase destroys the DNA, that DNA treated cells were not able to show the transformation. So the transformation can be inhibited with the mixture which contained the DNA. So from this they concluded the transformation agent is DNA. And they concluded that DNA is acting as hereditary material. Then later in the year 1952, two scientists, that is Hershey and Chase, they performed an experiment to prove that not only in bacteria, even in virus also, DNA is acting as the genetic material. So how they have proved DNA is acting as the genetic material in virus? So here they took the virus that affects the bacteria, that is named as bacteriophage, and the variety of bacteriophages, T2 bacteriophages. So they raised two sets of bacteriophages in that. And how can we grow the bacteriophages? We can grow the bacteriophages in bacteria. So here the bacteria they selected as E. coli. So they selected two cultures of E. coli. And the first one they named as A, the other one they named as B. In the first one, they took the T2 bacteriophage, in which they contain only two components, that is protein and a DNA. In this, the bacteriophage was labeled, the first set, the bacteriophage which is labeled as with P32, phosphorus, isotope of phosphorus, P32. And the phosphorus, since the phosphorus is a component of DNA, in that culture, in that E. coli culture, all the bacteriophages were, the DNA of the bacteriophages were labeled with P32 for many generations, in order to confirm that all the bacteriophages in that A culture, the DNA got labeled by P32. The same way in the other set, that is in the B culture of E. coli, they introduce the bacteriophage in which the protein coat is labeled. So protein means the main ingredient is sulfur. So the sulfur 35 labeled protein labeled culture, bacteriophages were allowed to grow in that second culture. And they conducted the experiment in 3 steps. So the first step is infection. So infection means the E. coli culture separately were allowed to infect with separately labeled T2 bacteriophage. In the first set, it is phosphorus 32 where the DNA is labeled. And in the second set, it is the sulfur 35 where the capsid was labeled. Then they were allowed to infect. After infection, the whole set were allowed to blend. So the purpose of blending means that is the whole culture medium along with the E. coli which contained the T2 phages. T2 bacteriophages were allowed to blend in a jar. So the purpose of blending was to remove the virus from the bacterial cells, the viral coats, the mixing, that is after a short time mixing the blender detaches the viral coats from the bacteriophages. Then after blending, the whole mixture were allowed to centrifuge. That is for separating the viral coats from the bacterial strain, because always the infection of bacteria is phage, that means viruses. When the bacteria is affected with virus, the DNA will enter alone, the capsid will remain outside. So this blending as well as the centrifugation was done to remove the viral coats, and by centrifugation settling of the E. coli and the coats will remain or will float in the top. So the steps they were following is, first step was infection, infecting, allowing the two different cultures of differently labeled T2 bacteriophage cultures were allowed to infect the E. coli. Then the second step, they were allowed to blend after some time to detach the virus from the bacteriophages. Virus means the coats. Then the separation of these coats with the bacterial culture is done by centrifugation. And after centrifugation, they observed the results. In that results, in the first set, they found out the radioactivity was detected. Most of the P32 in the pellet within the bacteria. So that is in the bottom. They were able to separate that one, and the bottom layer were showing the P32. And in the other set, the radioactivity was detected in the supernatant solution. So the supernatant solution means it is the protein coat where that is separated already. So in that the sulfur 35 was found out. So from this they concluded that even the virus, when they attack the bacteria, it is not the protein that is entering into the bacteria. It is actually the DNA from the virus is entering into the E. coli. And in order to prove that only, they have labeled the DNA separately as well as the protein or the capsid separately. Because DNA contains phosphorus, that is why phosphorus is labeled. Whereas protein consists of sulfur, that is amino acid which contain the sulfur. So there the sulfur got labeled. So from this they found out, or they proved that DNA is the genetic material. So this is the diagrammatic sketch. When for the exam, when they ask, you have to draw this diagram. So in that, the first one you see, that is bacteriophage. In this, DNA is labeled with P32. The other one, the sulfur. So here in this picture, just reverse only the sulfur. The first figure is sulfur. The capsid is labeled with sulfur. The other one, the protein is labeled. And in that, the sulfur radioactivity labeled in they detected after the experiment in supernatant, whereas the phosphorus radioactivity found out in the pellets which are deposited at the bottom. Now we'll move on to the conclusion. So here the observation: E. coli bacteria which were infected by radioactive DNA viruses were radioactive, but the ones that were infected by radioactive protein viruses were not radioactive. And the conclusion what they made: the resultant radioactive and the non-radioactive bacteria infer that the viruses that had radioactive DNA transferred their DNA to the bacteria, but viruses that had radioactive protein did not get transferred to the bacteria. Hence, DNA is acting as the genetic material and not the protein. So, in the bacteria or in the virus also, the genetic material, that is T2 bacteriophage. So, here the DNA is the genetic material. So, it is not the protein, instead the virus is acting, instead DNA is acting as the genetic material. Then the next concept, the DNA RNA world, which is the first genetic material. So when we look back into the evolution, in the evolution, it is believed that the primitive organisms, that is primitive cells we have to say, the primitive life forms, the first formed life forms, were carrying the genetic material which is in the form of RNA. Then later in the course of evolution, this RNA is replaced by DNA. So what makes this replacement, or what are the main reasons for why initially there was RNA for organisms and later why it was transformed or it is replaced by DNA? Was there any relation or was there any significance for that? So here, when we look into the atmospheric conditions of the primitive world, so in the earlier times, the atmosphere was anoxygenic. Primitive Earth was anoxygenic. So what do you mean by anoxygenic? Absence of oxygen. And it was reducing climatic conditions. And all volcanic eruptions, very hot climatic conditions, very unstable climatic conditions. All of a sudden, very hot, and the water was not there. Always the vapor formed the water, and it will be evaporated very fast, and they will be accumulated in the atmosphere. And suddenly it will condense together to form a very big cloud, and then precipitation will take place. Then again suddenly chilling effect, again hot temperature. So there will be a repetitive hot and cold climatic conditions. So in that climatic conditions, and that too in the reducing atmosphere and absence of oxygen, always favors an unstable molecule. So when we compare the DNA and RNA is more unstable. The reasons we'll explain later. And they prefer, in that course of evolution, they prefer always the presence of an unstable molecule which have the scope of modifying from one type to another to suit to the climatic changes. So that time the organisms prefer an unstable genetic material, and the preferable material was RNA. And RNA, when we study, it is very highly reactive because of the presence of OH group at the second position. And uracil is the which one? Uracil, the presence of uracil compound within the base, whereas thymine is more stable. Then RNA itself is made up of single strand. There is no complementary base pairing, so instability. Then pentose sugar, already I said, it is more reactive because second position oxygen is present, OH group. And they are showing catalytic activity. So what do you mean by catalytic activity? If any temperature changes, In order to survive in that enzyme activity of the RNA molecule can cut by itself. Then editing and all can be done very easily. Modification. As a result, the different types of combinations of characteristic forms can appear. So that unstable climatic conditions, organisms prefer the unstable molecule, which can always transform into new type of molecule. So in the initial stages or in the early stages, the life forms were fast evolving. They were fast evolving. They were able to modify in order to suit to the sudden changes in the atmosphere. But gradually, the appearance of or evolution of photosynthetic organisms, the appearance of chlorophyll molecules, evolution of oxygen as a result of photosynthesis, the climatic conditions also became very stable. Then the organisms prefer all the suitable conditions, all favorable conditions achieved, then the organisms prefer a stable component rather than an unstable one. The total atmosphere, the total earth, the condition of the earth has become very stable. Climate also very stable. Enough and abundance amount of oxygen. So from reducing climatic conditions, it has transformed into a stable. So that time, the stable molecule they preferred gradually, RNA molecule was replaced by DNA, which is more stable because they are double stranded and they are complementary base pairing, stacking of RNA base pair, nucleotide base pairs one over the other. Then the presence of thymine molecule, absence of OH in the one less reactive second carbon, that is H group. All this made the DNA more stable. And apart from that, the DNA molecule, when they replicate, the error chances were very less because if any error at the time of replication, immediately it will be repaired with the help of an enzyme, repair enzyme. So the mutation rate or the change that occurs in the DNA when it is replicating, they are very, very less compared to the RNA. RNA, there is no repair mechanism, whereas DNA, there is repair mechanism. So from one, when the cell is dividing from one set of DNA, when they replicate, the exact copy of the DNA is passed on to the next generation. Even organisms also prefer that stability because by the time they have established the whole, colonizing the whole entire earth with quasi atmospheric conditions, with very suitable climatic conditions. So they prefer no more change again. So they prefer more stable genetic material. So gradually from RNA, the genetic inheritance trait is passed on to the DNA. Now we'll move on to the next as an why when we compare the DNA and RNA. When we compare the DNA and RNA, which molecule is more stable, which molecule is less stable. Of course, we have learned previous topic that is RNA is less stable. Why it is so? What all characteristics of RNA, structural features, they makes them less stable when compared to the DNA. The RNA is single-stranded polynucleotide chain. There is only one strand, so there is no possibility of complementary base pairing. Then the second point is there is base pairing is not occurring, hydrogen bonding is not occurring between them, so that also is second point. The third point, at the pentose sugar, the 2OH group in every nucleotide makes them more reactive. OH means always they will have a tendency to bind with other components. And the fourth point, the base sequence, when we are saying the base sequence, the uracil is present in RNA, whereas methylated uracil, that is thymine. Thymine is methylated, that means the free group is methyl, it is bound with methyl. So methyl group methane is less reactive. because all their carbon, the valencies are satisfied with hydrogen. So then it is more stable. Thymine is more stable compared to uracil. So the uracil carrying RNA compared to thymine carrying DNA, it is less reactive. It is more reactive. The RNA is more reactive. Then RNA itself, the 23S RNA in prokaryote or 28S RNA in eukaryotes, when we are saying they are showing the ability of catalytic. That means they are one component, this component can show the ribozyme activity, which is an enzyme. So the RNA, whenever they feel if any editing is to be done, it can edit by itself. It can cut its own molecule into fragments. So when we discuss about the uniqueness of a species, the genetic material must be stable. It should not often change. So organisms, they prefer always stable genetic material. So RNA, since they are showing their catalytic activity, it is less reactive. Then the last one, since they are able to change their base sequences by cutting and editing and all, it will become that by nature itself it will become unstable and it can mutate and evolve faster. So the organisms, when the climatic conditions and all, they prefer more stable. So DNA. So all these points can be given as why DNA is more stable compared. So when you are discussing why, the question is asked why the DNA is more stable means when you have to write it another way, because the DNA strand consists of two polynucleotide strands with base pairs, complementary base pairs. And in each one, the steps, it is arranged in the form of a spiral helical structure which is more stable. That structure itself gives them additional stability. And in that, the base pairs are arranged one over the other. That too, they are bound with hydrogen bonds. Then, in the second position, less reactive group, that is only H group, only is present. Then presence of base, that is thymine, which is methylated uracil. And DNA does not show any catalytic reactions. Hence, the DNA is more stable. It cannot mutate faster. And in DNA itself, there is proofreading and repair mechanism. Proofreading and repair mechanism when they undergo the replication. So, which resist the changes. All these points you can write it. Now, when we discuss what are the minimum requirements for a particular molecule to be called as the genetic material. So here there are four qualities, four characteristics which can be satisfied for both DNA as well as RNA, not for the protein. So three components I said: DNA, RNA and protein. So why the protein is being taken out from the genetic material? Because RNA, few organisms, RNA is acting as the genetic material. And majority of organisms, DNA is acting as the genetic material. Why it is so? What are the minimum requirements for a particular molecule to be called as the genetic material? So the first point, it should be able to generate its own replica. So in this aspect, when you are saying DNA, we know that DNA can reproduce itself, can make its own replica. So what about RNA? In virus, RNA, even though they cannot directly make RNA, they can make a complementary DNA. From that they can make an RNA. Okay, so DNA as well as RNA can make its own replica. Then it should be stable and chemically and structurally, because their structure should be stable. So in DNA, it is most apt for the DNA. DNA is structurally, they are helically wound with the help of two polynucleotide strands. And chemically also, the components in the DNA are more stable. We learned that thymine and pentose sugar with a one O less. That means H group alone. So it is more stable. The third point, it should provide the scope for slow changes that are required for evolution. So even though I said they resist the changes, but proofreading and error. But even though occasionally, not often, occasionally in the DNA replication, error can occur. That is, every 10,000 base pairing, there is the chance of uh, one error. That is, the error rate of DNA replication is 1 in 10 to the power of 6. One error in 10 to the power of 6 base pairing. So there can be occasionally very minor changes can occur which can lead to the evolution. The fourth one, it should be able to express itself in the form of Mendelian characters. So what does that mean? When from one organism into another organism, when they transform, when they inherit, the characters should be able to transmit to the other organism. Inheritance, it should inherit the DNA from one organism into another. That only in the reproduction we have learned when the gamete is forming. The gamete forming cells, the myocyte will pass on that chromosomes, the haploid set of chromosomes to the gametes. This gametic fusion only results in offsprings. So always the DNA can pass from one organism to another. And as a result, the characteristics can be passed on. The traits can be passed on generation after generation. So all these are the important characteristics. Then all these points we have discussed why RNA is highly reactive than DNA. All these points, the RNA nucleotide, the additional OH group, RNA shows catalytic activity, hence changes often. Then uracil is less stable as compared to thymine because thymine is methylated, uracil. And the RNA itself is labile and degradable, very easily it can be degraded. Then RNA is single-stranded because absence of base pairing. Then RNA molecule itself is less stable, it will mutate even very fast. Therefore, among the two nucleotides, DNA is better genetic material. Then later, the scientists they inquired about how the DNA is replicating. So when the cell is dividing into two, how the exact number of DNA is maintained. So when one cell they contain, in human cell means they contain 46 chromosomes. So from that, when the cell is dividing into two, each daughter cell will get the exact number of DNA or the chromosomes. How they are getting the exact number. So this concept, the replication of DNA, how it is done, the two scientists, that is Watson and Crick, they propose that when dividing, the DNA is adopting a method that is called as semi-conservative mode of replication. So what do you mean by semi-conservative mode of replication? Means they said whenever from a DNA molecule, when they transform into two molecules or when they divide into two molecules, since the DNA molecule consists of two strands, here in the figure it is given, two strands, one is with blue, the other one is with red. So when they replicate into two, in that process, the two polynucleotide strands will separate and each polynucleotide strand will act as the template. And based on that template, from the medium, they will take the base sequences and they will form a new strand. And as a result, two nucleotide strands will form. Out of that, one nucleotide strand will be the parental strand and the other one will be the newly synthesized strand. So here the parental strand is conserved in the new daughter DNA. That type of replication is called as semi-conservative replication, mode of replication. So this theory was proposed by whom? The Watson and Crick. But they couldn't establish any experimental proof. Later, the experimental proof was given by the scientist. That scientist names were Messelson and Stahl. What they did is they allowed to grow the E. coli, the bacteria, in a medium which contained isotope, heavy isotope of nitrogen, that is N15, and the medium which contained ammonium chloride. In that the nitrogen was labeled as N15. And for many generations they cultured the E. coli in the same medium, that is heavy isotope containing ammonium chloride solution. So the E. coli, as it multiplies, the DNA also multiplies, and they divide and again they multiply. So after many generations, they took a portion of that sample and they have undergone a special type of centrifugation that is called as density gradient centrifugation in a solution, that is cesium chloride. So when they extracted the DNA, they were able to separate the DNA in a particular range, that is intermediate density. Not intermediate, in the bottom range, that is heavy. Since this one is heavy, the DNA were deposited at the bottom region of the certificate of the test tube after the density gradient centrifugation. So from that they understood that the DNA, they took the nitrogen N15 from the medium and all the DNA was labeled with N15. Then after that, he transferred this N15 grown culture, a group of that culture, into another medium which contained normal ammonium chloride. That means N14, which is light. Compared to N15, it is light. And every 20 minutes interval, they took the sample from that and they were undergone the density gradient cesium chloride centrifugation. And when they took the sample after 20 minutes and they have undergone the centrifugation, they have found out that after 20 minutes, the DNA, when they extracted, they separated, they found they were deposited at the middle region of the test tube. And from that they concluded that the DNA, the density of the DNA is intermediate. It is not light as N14, it is not heavy as N15, but instead it is in between the range of N15 and N14. So they confirmed that the DNA, one strand will be N15 and the other strand will be N14, and they named it as hybrid DNA. Again, after second generation, that is first generation, second generation, third generation, that is after 40 minutes when they extracted from the culture which was grown in N14 itself, they extracted and they separated the density gradient centrifugation in cesium chloride. They found out that two sets of DNA extraction, that is one set was in the intermediate density and other set was deposited at the top. So the top, which one was deposited, it was light and both the strands were N14 absorbed, and the intermediate were hybrid, that is one strand was N15 and other strand was N14, and they were not able to isolate any heavy type of DNA. So from this they concluded that the replication, when the DNA is replicating, out of these two polynucleotide strands, both the strands will separate as it is. And each strand of that one will act as the template. And by reading that base sequences, the complementary base pairing will take place by absorbing the bases from the medium. So that as a result, one strand of the DNA, newly formed DNA, will be template or the old strand, and the other strand will be the newly synthesized strand. So here the template is retained in the next generation also. One of the template strand is retained In the newly synthesized, that type of replication is called as semi-conservative mode of replication. More or less the similar type of replication method they have adopted even in plants also. It is done by the scientist Taylor and Vishia Faba. Faba beings will say, and there also they demonstrated that even in plants also they are adopting the semi-conservative mode of replication. So with this I am concluding today's revision. In the next class we will discuss the topics after this. Thank you very much.